Hello, everybody. I am going to give you some introduction into protein metabolism and ultimately amino acid metabolism. The other day, we focused on amino acid biosynthesis. Today, we're going to be focusing on breakdown of amino acids. But before we get into the breakdown of amino acids, we need to get into the breakdown or the catabolism of proteins. And so protein metabolism, protein catabolism, specifically the breakdown of proteins, it serves a couple of different roles within a cell. One role is that it can be used to remove aberrant or abnormal proteins. So if something has been, misfolded or something like that, protein turnover or a protein can be broken down. Another reason is to deal with or manage the nitrogen within a cell. Because one thing that we'll see fairly soon is that nitrogen containing compound like ammonia can ultimately be toxic. And then the third and maybe most, I would say most significant or most I would say most significant, although maybe not the most relevant to this discussion or this conversation, is that when a protein is broken down or when a protein is catabolized, well, it's no longer in the cell. And so whatever that function that protein was doing has in theory been removed from that cell. So proteins can be catabolized for the purposes of regulating that function. So there are proteins that are regulatory, which have often a very short half-life. So it's going to be synthesized and then it's going to be broken down. Now, the ways in which a protein can be degraded is that it could be degraded in a eukaryotic cell in the membrane-bound organelle known as the lysosome. Now, lysosomal degradation, one thing that's important to recognize, and it seems fairly, or I hope that it's fairly apparent, is that this is a breakdown that's taking place in a membrane-bound organelle. So what that means is that a protein has to be transported into the lysosome. Now, lysosomal degradation, I think that the players involved are not so different from players that we've talked about previously. It's not like this is just some sort of garbage disposal and everything just gets dumped in there. Instead, there are proteases that are unique and localized in the lysosome. These are proteases that will function at a pH of approximately 5 because the pH within the lysosome is approximately 5. So a lysosomal protease, and just to kind of review the definition for a protease, that is an enzyme that breaks peptide bonds. So proteases, there are specific proteases, lysosomal proteases, that will function at a pH of 5. Now, one thing that I want you to recognize is that I'm saying this lysosomal. I'm using the phrase lysosomal protease. And I'm going to underline lysosomal because of the fact that, well, this is a protease or these are proteases that are found in the lysosome. What's implied there is that there are proteases in other areas of the cell. There are cytosolic proteases. There are probably mitochondrial proteases, chloroplast proteases in the event of a photosynthetic organism. But I just want to kind of clarify that. Now, in a well-nutrient or a well-nourished or a nutrient-rich environment, these proteases are going to be non-selective. And that means that if you present them with 10,000 different unique proteins, it's kind of flipping a coin which one it's going to go after. That protease isn't going to have any selective criteria. It's not going to say I have to degrade the 50 amino acid protease or protein before I do the 70 amino acid protein. Under starvation conditions, however, and I want you to think about this as, well, under starved conditions, that cell is really seeking out some sort of nutrients. And so what it's going to do is it's going to say, effectively, what are the targets, what are the proteins that I can degrade and not undermine my existence? What are the easiest things to degrade? And under starved conditions, lysosomal proteases are going to look at proteins that have a KFERQ polypeptide found within them. So what that means is that if we take, here's our N termini all the way to our C termini. Here's our polypeptide. In lysosomal protease or protein degradation, let's say that we have two different proteins, protein A and protein B. Now, under well-nourished conditions, the proteases proteases found in the lysosome, whether they pick A or B, it doesn't really matter. They're not going to be very selective. Now let's imagine that protein A has this KFERQ peptide. Okay, so A has that KFERQ peptide, B does not. Under well-nourished conditions, it's flipping a coin. The proteases within the lysosome, it's not going to go after one more than it goes after the other. It's going to degrade them both, and it might go after B before it goes after A. It might go after A before it goes to B. It's just a flip of a coin. Whereas under starvation conditions, these lysosomal proteases, they're going to go after protein A first, because the proteins that have this KFERQ, think about those as you don't have to have them a little bit in order to continue functioning. The way that I always kind of think about it is, let's say that here's a house. I've drawn a house here. And actually, I'm not a good enough artist to really define this any further. But the way that I think about it is you have top-down view of a house. You've got your walls here, your exterior walls that serve as supports for your roof. Then you've got these interior walls. Let's say here's an interior wall. Under starvation conditions, the proteases, I'm sorry, are going to go after these interior walls. Because if they take out the interior wall proteins, you know what? The roof is still going to stand up. Once it gets through all of those, if nutrients are still needed, sure, then they'll go after these exterior walls, but those are going to be kind of the lower priority ones, just because, well, that's like things are crashing down. The cell needs these nutrients in all situations, or it absolutely needs them more than anything else. So this is the case in the liver and the kidney. This is not the case in the brain. I think that it's important to understand the difference between starvation conditions and non-selective under well-nourished conditions. So lysosomal activity and the activity of those proteases is going to vary. And lysosomal proteases tend to be a little bit more active in certain situations. For instance, in arthritic or people afflicted with arthritis, they might have more or larger lysosomes within their cells. They might have more lysosomal proteases, in which case they're breaking down proteins at maybe a little bit of a higher rate than someone who is not. So lysosomal degradation is localized to a membrane-bound organelle. That organelle, you have to put the proteins in order for them to be broken down. Another method or another way in which a cell can break down protein is using something known as a proteasome. So proteasome, I want you to see that OME and think about genome, proteome, microbiome. These OMEs are kind of global views. So the proteasome is a large protein complex. And the way that I always kind of think about it is it's almost like a paper shredder. It's your garbage disposal. This is an ATP dependent protein complex that has two 19S caps and one 20S proteasome. So these 19S caps, first of all, the S is a measurement for something known as analytical ultracentrifugation. S stands for Svedbergs. And 19S has to do with the sedimentation coefficient. So basically, where something separates from other components when you're doing analytical ultracentrifugation. The key is that this is a structure that basically has two caps and one kind of functional unit in the middle. So that's kind of the takeaway from this. Now, the way that this functions is that these 19S caps, they select and they unwind ubiquitinated proteins to be degraded, sorry, not degraded. So what they will do is, let's imagine we've got a polypeptide out here, polypeptide out here. And these 19S caps kind of serve as almost like baits, and they grab onto something known as a ubiquitinated protein. Another way that, or at this point, you could think about them as they're looking for a protein. They're going to grab onto a protein. What they're going to do is they're going to unwind it. So that means that this 19S cap has the ability to disrupt a number of different, they have the ability to disrupt intermolecular forces. So those attractions between different R groups, this 19S cap or these 19S caps are able to disrupt those. Now, this next thing, this ubiquitinated, we're going to talk about that in just a little while, but another way that you could think about that is the 19S cap selects and unwinds tagged proteins. So, these tags are ubiquitin subunits that have been added to a protein, almost targeting it for degradation. So the 19S caps, they grab onto the proteins and they basically unwind them. They take whatever structure they have and they make them a linear chain. Those linear chains are then fed into 20S proteasome. So it's almost like this protein right here, protein A, well, it's going to be picked up by the 19S cap. And when it's picked up by the 19S cap, it's going to begun being unwound and processed into the 20S proteasome, where this 20S proteasome is going to degrade it, break it into, chop it into little pieces. These pieces are going to be approximately 8 amino acids long, and those 8 amino acid long pieces will also be separated from their tags. Okay, so in order for the proteasome to function, a protein has to have multiple ubiquitin tags added to it. So the proteasome is a cytosolic protein. This is a protein complex contrary to our lysosomal degradation. We were talking about a membrane-bound organelle. That membrane-bound organelle, well, it sucks in a protein and, well, it processes it. Whereas the proteasome here, what it's going to do is grab onto a tagged protein, and it's going to slice and dice that protein into smaller pieces. Those pieces are 8 amino acids long. Now, those 8 amino acid long fragments, those short polypeptides, those are ultimately going to be processed and broken down further into individual amino acids and some much shorter peptides, shorter than 8 amino acids, but those will be broken down and those will undergo our amino acid catabolism. Now, ubiquitin, that's the important thing that I want you to get into a little bit. Ubiquitin is a tag. Ubiquitin marks a protein to go to the proteasome. So any random protein, which I'm going to draw right here, and I'm going to label the N termini and the C termini. This random protein here, well, it's not simply going to appear at the proteasome's front door and say, all right, I'm ready to be degraded. Instead, this protein has to be ubiquitinated. And what that means is that it has to be modified by the addition of a ubiquitin subunit. So here's our protein that's going to be ubiquitinated. Now, ubiquitin is a 76 amino acid protein. It has a sequence that is very highly conserved across organisms. And think about it as it is itself a protein that doesn't have a particularly unique enzymatic function or anything of that nature. Instead, we treat it almost like it is a tag. And one of the things that it can look like is, here's our protein right here, and here's a little ubiquitin subunit that's been added. Here's another ubiquitin subunit, here's another, and here's another. So, excuse me, this is an example of a protein or a model of a protein that's been polyubiquitinated. 4 individual ubiquitin tags have been added to this protein. Now, what is important to know that not every single protein gets ubiquitinated. Instead, there are some things about the sequence that the enzymes that are involved in ubiquitination will recognize. So the enzymes that are involved in ubiquitination, what they'll do is they'll see, here's 3 different proteins, A, B, and C. And they'll say, okay, we're going to look at the sequence for A, we're going to look at the sequence for B, we're going to look at the sequence for C, and we're going to see if it meets what we're looking for as a protein that we need to add a ubiquitin to. And what they're going to do is they're going to look at the N termini. And the N-terminal amino acid is a very important kind of factor or a very important kind of signal of sorts. And what they're going to look at is basically, is there a specific sequence that will cause them to very quickly add a ubiquitin subunit. Depending, if you compare certain proteins and terminal sequences, there are some that are correlated with a protein that's degraded very rapidly. And then there are others that are correlated with a protein that it stays around for more than 10 hours. And one of the signals that's common amongst proteins that get degraded very quickly is following sequence, which is PEST. That's a proline followed by a glutamate, followed by a serine, followed by a threonine. If that PEST is, let's say that that's present in protein B here. It's not in A and it's not in C. Well, the enzymes that are involved in ubiquitination will see protein A and they'll say, not interested. They'll see protein C and they'll say, doesn't have a PEST sequence, not interested. Ah, but B does have that PEST sequence. So you know what we need to do? We need to ubiquitinate that. We need to add a bunch of these small 76 amino acid proteins onto protein B. And when we add a bunch of those ubiquitin subunits onto protein B, that's going to basically trigger that to be degraded. That protein B will get degraded. Proteins A and C up here, since they didn't get ubiquitinated, chances are it's going to take a long time for them to be degraded. Now, ubiquitination is a very tightly regulated process because the proteins that are involved in ubiquitination, they're not necessarily going to be active all the time, because if you have a protein that basically is going to get degraded, you want it to happen at a certain time. You don't want it to happen at all times during the cell's life cycle. Instead, there are sometimes maybe there's something with like a circadian rhythm that, okay, well, now is when these ubiquitin or these enzymes that are involved in ubiquitination become active, so then they can target these proteins for degradation. There's any number of other sort of stimuli that can lead to that. Now, there are three classes of the enzymes that are involved in ubiquitination. There are three classes of these ubiquitin ligases. There's the E1, E2, and E3. Excuse me. Now, what E1 is going to do is it is basically going to take this ubiquitin subunit and more or less prime it, ready to be added onto the protein that we want to degrade. And it's going to do this in an ATP-dependent fashion. It's going to take the hydrolysis of ATP to AMP and more or less bind this ubiquitin subunit to itself. So in step one here, what we have taking place is the C termini of our polypeptide, of our ubiquitin polypeptide. And we have this SH, which would be indicative of a cysteine residue. We have this, E1 is our enzyme that has a cysteine residue that we are going to more or less bind our ubiquitin to. Okay, so we end up with this molecule right here, E1 with that cysteine residue with our ubiquitin covalently linked to it. The next thing that we're going to do is this E1 is going to transfer ubiquitin onto another enzyme known as E2, resulting in this molecule right here, where we have a E2 ubiquitin. So a way that you could think about this is E1 is E1 activates ubiquitin and passes it off to E2. E2, when it is ubiquitinated, is ready to transfer the ubiquitin onto our protein that we're going to actually be condemning or more or less degrading. The enzyme that does the activity of transferring ubiquitin onto a target protein is E3. These are all enzymes that are known as ubiquitin ligases. And what they're going to do is basically activate ubiquitin, which is commonly written as UBQ. It's going to activate ubiquitin. It's going to transfer UBQ to E2, which I personally think about E2 as kind of a gatekeeper. Because when you think about E1, E2, and E3, E3 is the enzyme that says, all right, ubiquitin on an enzyme, on a ubiquitin ligase, I'm going to take that ubiquitin and put it onto our enzyme that we want to degrade. Well, we're taking it from E2. I kind of think of E2 solely as like a gatekeeper because E1 is there to activate it, but it also forms a covalent link between itself and ubiquitin. But then it hands it off to make a covalent link between ubiquitin and E2. It's the E3 that takes the ubiquitin from E2 and puts it onto that protein that we're going to degrade. Now, one thing that's important is this E3 ubiquitin ligase, when it takes ubiquitin from E2 and puts it onto the protein that we're going to degrade, that protein that we're going to degrade absolutely must have a lysine residue. Now, if you remember on the previous slide, I mentioned a PEST sequence. Well, that's just one polypeptide that is in the N-termini of a protein. None of those residues are necessarily what we're adding our ubiquitin to, but they are relevant for kind of identifying proteins that will be degraded. But ubiquitin is going to be added to a lysine residue. So another way that you could think about this is if a protein does not have a lysine, it's not going to be a target for ubiquitination. But at the same time, if you think about a protein that's 150 amino acids long, it's going to have probably at least one of every single amino acid, and it might have a higher constant or a higher number of some amino acids than others, but chances that it has a lysine, or chances that any random protein has a lysine, they're pretty good. But that is kind of your minimum criteria. You have to have a lysine residue in order to get ubiquitin added to it. So at any rate, this is the process of ubiquitin or ubiquitination. You have to activate that 76 amino acid long polypeptide, or that 76 amino acid long protein, that ubiquitin. Add it to E1. E1 transfers ubiquitin onto E2. E2 then serves as the kind of conduit or the gatekeeper that E3 can take ubiquitin from E2 and put it onto our protein that we're going to degrade. Now, ubiquitin is a very dynamic process, or it's very dynamic in the sense that it is not necessarily like, let me rephrase that. Ubiquitins can be added to and removed from a protein. The bond that's formed between our ubiquitin and a lysine is what's known as an isopeptide bond, which is what we've got at the bottom here. The isopeptide bond. We can break that bond with an enzyme known as an isopeptidase. So the addition of a single ubiquitin doesn't necessarily condemn a protein to be degraded. Instead, it's almost like I've marked it, or I put the first mark on for this protein to be degraded. Is it going to be degraded? Not necessarily, but I've at least marked it so that it's possible that it will be. But that ubiquitin can be removed. So I always kind of think about ubiquitin as something that it can be added, to more or less like slow down a protein's function? Or are the conditions right where I need to degrade this? I'm going to tag this with a single ubiquitin and store it over here. I might come back to it to degrade it, but for the time being, I'm just storing it over here. So a single ubiquitin can serve one purpose, but proteins that are going to be degraded are usually going to have multiple ubiquitin, and those ubiquitins are generally going to be found in a chain. So what I'm looking at here is, I'll show kind of the different interpretations here. There's our polypeptide, and to see, this isn't drawn at all to scale, but instead, here's the ubiquitin, and so this is actually a 4 ubiquitin chain. So 1, 2, 3, 4. So this protein got ubiquitinated in one place, and then a chain of 4 ubiquitins was added to it. Actually, you know what? That would be a better, I'm going to erase this, because this would be a better model for polyubiquitination. Multiple ubiquitins would look a little bit more like this. And here's the ubiquitin. Here's the ubiquitin. So there could be multiple individual ubiquitins that have been added to the polypeptide, or there could be a chain of multiple ubiquitin subunits added. Now, I think the important thing that you know is that these ubiquitins can be removed. But if they're not moved, oh boy, sorry. If they are not removed, well then that protein has basically been targeted for degradation, in which case it's going to go to the 26S proteasome, which has two 19S caps and one 20S proteasome, one 20S kind of shredder central unit. So those caps will identify one of these guys, either this one or this one. Those caps will grab onto those proteins and unwind them, thread them into the 20S proteasome where they'll be diced up, and those diced up pieces will be released as 8 amino acid long peptides and ubiquitins. So I've always kind of thought that the ubiquitins are almost like a bait and switch. Like they say to the protein, hey, I'm just going to grab onto you, and we're just hanging out. And then they go to the 26S proteasome, breaks up the protein. The ubiquitins are unharmed. They're released as individual ubiquitin subunits. And the protein is degraded into 8 amino acid long polypeptides. So I'm going to go ahead and stop right here, and just to kind of break things up and continue in the next video.